Okay, hello everybody. This is Paul Sims once again. I'm the chairman at Pharma, and I'm very pleased to welcome you to today's webinar. You can see some smiling, happy faces on your screen, who I shall introduce you to in the moment. But you can see that the name of today's webinar is the RWE Regulatory Evolution: How to use RWE to augment RCTs and advance the drug development paradigm. That's quite a mouthful, but I think that you can all tell that this is going to be a very interesting subject. In fact, nearly 1,500 people have signed up, so it definitely is an interesting subject. And I personally believe that we're going to be talking about evidence evolving at a rate faster than we've seen in the past 30 years at least, and it's a very exciting time. Real-world evidence is having an increased impact on the development and commercialization of new treatments. And the FDA, prompted by the 21st Century Cures Act, is engaged in a process to determine exactly how RWE can augment or even substitute randomized clinical trials. But with, of course, the enduring goal of getting the right treatment to the right patient faster, but still correctly and still safely. So today we're going to be talking about exactly this. We're going to be hearing more about RCT DUPLICATE, which is an FDA-funded demonstration project in which real-world data is used to replicate the findings of 30 completed phase 3 and 4 randomized controlled trials and to predict the results of 7 ongoing phase 4 trials. So we're going to hear a little bit in terms of some of the findings, and we're going to determine how you in pharma companies can apply some of the learnings from this with an honest assessment of the effort required to have an impact. And as you can see, we really couldn't be joined by a better group today in terms of leading this conversation. Let me introduce them briefly to you. So Dr. Sebastian Schneeweis, sorry, I knew I'd screw that up. Schneeweis is the Professor of Medicine and Epidemiology at Brigham and Women's Hospital and also at Harvard Medical School. His research is supported by NIH, PCORI, and FDA grants. He focuses on the comparative effectiveness and safety of biologics and pharmaceuticals. He's developed several analytic methods to improve the validity of epidemiologic studies using complex healthcare databases, particularly for newly marketed medicinal products, and bundling these into a rapid cycle analytics framework. Doing some very exciting work, and he is a co-founder of Aetion, who I will talk about in a moment, who've been very supportive on this webinar. Jeremy is a co-founder and president and chief science officer at Aetion. He's one of those rare breeds. He has an advanced qualification in both computer science and epidemiology, and he, as I said, was a co-founder of Aetion back in 2013, and he actually leads Aetion's efforts to design, scale, and communicate scientific solutions and methodologies for obtaining valid and timely medical evidence from real-world data. So I said I'd mention Aetion. Aetion have played a key role in making sure today's webinar happened, in actually helping to design and create it. They've been an excellent partner to work with in getting this very popular topic out into the open, and I'm really very grateful to Aetion for that. Stephen, next to Jeremy, he actually heads the Global Safety and Pharmacoepidemiology Group at Eli Lilly, and he's been there for 11 years. He's now in this role responsible for all RWE studies pertaining to safety and pharmacovigilance. So excellent that you could be with us, Stephen. Thank you. And last but certainly not least is Brian Bradbury, who is the Executive Director at the Center for Observational Research at Amgen. He's been with Amgen now for 15 years, and he's also an adjunct assistant professor of epidemiology at the UCLA. And he leads a team of epidemiologists, biostatisticians, data scientists, and programmers responsible for Amgen's RWD platform. And it's actually a platform which is used for a variety of essential functions, both pre and post-market. So we have a very, very good group, and we indeed have got a very solid audience. In fact, I'm being prompted on my screen as I'm talking to me that lots of people are trying to join and hitting an upper limit even. I apologize to those people. And of course, we are recording today's session, so there'll be the opportunity to listen back, to share with colleagues. But to those of you who are here, and who indeed have made it. Let's make this not a one-way conversation. Let's make this a dialogue. And if you look at the right-hand side of your screen, you'll notice that there's a questions box. Perhaps you could write a quick hello in there to me right now so that I can tell you haven't fallen asleep yet, and so that I know that you can hear me. So, yep, I can see some hellos coming in. Thank you very much. So appreciated that. So now that you've still awake, yes, good. Now that you know where that is, please do use it throughout so that we can get a good conversation going. But the first thing I'm going to do on this webinar is actually ask you a question. So if you look at your screen right now, I'm just going to bring up a question. When does your organization use RWE today? And obviously, if you're not from a pharmaceutical company, I'd like you to answer this on behalf of clients you work with. And if you're not from any kind of agency or network, for example, you're a patient, then perhaps this question isn't for you. But when does your organization use RWE today? And by this, I mean in a significant manner, of course, not just pilots, but using it in a comprehensive way. So this is the only question we're asking today, which you can select multiple options. So drug discovery, preclinical, clinical development, regulatory, market access and HEOR, launch marketing, and post-marketing safety and regulatory response. Okay, so I've got about half of you voted. I'm just going to hold that open another 3 seconds. 3, 2, 1. Okay, glad to see so many of you voting there. Let's have a look at the results. Okay, so what can we say from this distribution? Well, clearly there's a winner, market access, probably no surprise, nearly 3/4 of you, and 2/3 of you choosing post-marketing safety and regulatory response. The least on a drug discovery and preclinical basis, but still a significant minority. Okay, so that perhaps tells you where we are, a little bit of a benchmark for everybody there in terms of where we are today. But before we move on, I'm afraid your work is not done. There is another question for you. So if you look at your screens, this one I think could be a very interesting one indeed. Oh, sorry, I've just realized this is the wrong poll. I apologize. That was not the intention. Here we go. Sorry, have a look at this one. 10 years from today, how will the ratio between numbers of RCTs and RWE have evolved? So in other words, are we going to see a big change in how many RWE studies are conducted relative to clinical trials? So the top option on this list is pretty much as we have it today. RCT is still the gold standard, supplemented in some cases by RWE. Or we're going to see RWEs as the gold standard, supplemented by RWE in the majority of cases. Remember, this is 10 years from today. Third option is equal importance. So RCTs and RWE, probably the same number of studies being done in both 10 years from now. Or we're going to start to see it go the other way. RWE now the most common. RCT is no longer used in every treatment or potential treatment. And then finally, RWE is now dominant and RCT is actually used in a minority, less than 50% of new evidence cases. So where do you think it's going to be 10 years from now? It'll be interesting to see where we go. We've got about 62% of you voted. I'll open another 3 seconds. 3, 2, 1. Thank you very much, everybody, for voting. Let's have a look at the results. Okay, so we've got a bit of a normal distribution with a bit of a skew towards the upper end. So most people not believing that it's going to change a huge amount from where we are today within a 10-year time frame. Perhaps over the course of this webinar, some minds might be shifted. Maybe we should even ask this question again at the end and see what happened. But yeah, I'm going to just park this result for the moment. We might come back to it and discuss it later, because the next thing to do is to introduce the first set of panels, which are going to be shared by Jeremy and Sebastian. So Jeremy and Sebastian, I'm just going to hand over the screen to you now, and perhaps you can take us onto the next stage.
Thanks so much, Paul. Getting the screen going here.
There we go.
Great. And so, Paul, thanks so much. And this is Jeremy Rasson, co-founder, chief science officer, and president at Aetion. I'll speak for just a moment and turn it over to Sebastian Schneeweiss. As you mentioned, next slide, the topic here is the real-world evidence regulatory evolution. Why are we talking about this? Next slide. We're talking about this because the regulators are talking about this. So I wanted to share two quotes, one from EMA, one from Scott Gottlieb shortly after he left FDA. And I'm just going to read these to you because I think they set really good context for the discussion that's upcoming. So real-world data hold the promise to substantially increase the effectiveness and efficiency of all processes in the development and utilization of medicines, from research and development, that's maybe that first poll question, to regulatory decision-making, pricing and reimbursement decisions, to use in medical practice. FDA has a perspective as well. So as I said, Scott Gottlieb, a major cultural shift is underway at FDA when it comes to the fundamental framework of the drug review process. Sponsors should take note. The changes enable FDA to fulfill a holy grail of drug review, enabling more widespread use of practical data gleaned of routine clinical practice to provide greater insight into drug efficacy. And it's a really interesting article. I recommend afterwards that you click the source link at the bottom. That gives us a little context. I'd now like to turn it over to Sebastian Schneeweiss in just a moment, who will be talking about the RCT DUPLICATE demonstration project, as Paul mentioned. And then after a little bit of discussion, I'll come back and talk about regulatory precedent and the opportunity here for pharma. So Sebastian?
Sure. Hello, everybody. This is Sebastian Schneeweiss. Next slide, please. I'm going to tell you a little bit about a demonstration project that is part of the FDA's effort to fill in the real-world evidence framework. By 2016, the 21st Century Cures Act mandated FDA to develop a guidance for industry that clarifies FDA's use of real-world evidence for regulatory decision-making. And by the end of 2018, they released this framework document that outlined their current thinking and what they want to achieve for the next 3 years. And by the end of 2021, they will provide a guidance for industry. When you open this framework document, you will see two important definitions. FDA defines real-world data as data generated from the healthcare system and that are routinely collected from a variety of sources. So two aspects are important: routinely collected data and from a variety of sources. And they're quite open. They're thinking way beyond claims data, electronic health record. They're thinking wearable devices. They're thinking patient-reported outcome, collecting the measurement tools. So they're quite generic here. And they define real-world evidence, then, the clinical evidence that you derive from such data, from real-world data, to quantify the potential benefits and risk of medical products in order to build decisions on that. Next slide, please. And when you open this document, there are really 4 sections on this. There's an introduction, kind of outlining what the scope is of this. And then comes the section telling the reader what they have done already and how they are using real-world evidence routinely in today's federal FDA that is largely on the safety of medications, that is in rare disease settings and things like that. The 3rd chapter is where they outline where there is still a knowledge gap and where they want to push the envelope. That is using trials, pragmatic randomized trials. We have baseline randomization, but you use real-world data to collect outcome information, and how can we improve such trials and the knowledge derived from that. A lot of text on assessing the fitness of real-world data to support effectiveness decisions, the fit for purpose. What does make real-world data good enough to base important decisions on it? There's a lot of text on study design options and how to best design these non-interventional studies in order to derive knowledge that you can, as a regulator, make decisions based on. And then we talk about data standards, data provenance. How can we make sure that we fully understand the data that are routinely collected that gave rise then to the evidence analysis, the effectiveness analysis. And they have a block on stakeholder engagement. These are where they talk about demonstration projects, one of which I will illustrate to you. But they also very sincerely, I think, ask for everybody in the community, that is everybody on this call and way beyond that, to participate in this effort to better understand how real-world evidence can support regulatory decision-making. Next slide. And on the European side, there came a similar document out, Regulatory Science to 2025. It's a very worthwhile read, and it comes up with 5 key domains catalyzing the integration of science and technology and medicine development. They talk about real-world evidence in this point number 1 right away. Number 1 is number 2, deriving collaborative evidence generation. They talk about evidence networks, real-world data networks, and how that can help inform regulatory decision makings by the EMA. And way down there, number 5, enabling and leveraging research and innovation in regulatory science. How will you then connect what you learn from real-world evidence to the regulatory decision-making process? So all these challenges that the FDA has identified are almost mirror imaged in the EMA report 2025. Next slide. So it's really, we're talking a worldwide phenomenon. Let me now dive into these 2 projects that are funded by the FDA under the umbrella called RCT DUPLICATE. The main project is to identify 30 RCTs that lend themselves to be reproduced using real-world data to generate real-world evidence, and then compare the results between the RCT and the real-world evidence. And that study was extended by a second study funded by the FDA to focus on 7 ongoing phase 4 randomized trials and predict the outcome of those trials while they're still ongoing, and prospectively make an assessment based on real-world data and real-world evidence, and then compare whether the prediction got close enough to the randomized trial evidence. The overarching goal is to identify a space in terms of indications of outcomes of study questions, of effectiveness questions, where we have confidence that non-randomized designs using healthcare databases can support regulatory decision-making. This is about building confidence. This is not about saying that every real-world evidence study needs to be backed up by a randomized trial. Far from that. And the FDA recognizes that with real-world evidence, you often learn different things. We learn more than in randomized trials. But at some point, and that's the main goal of this study, we need to calibrate. Can we, in these situations, can we come to the same conclusion given certain design analytic choices? And can we also develop a process that the regulators will agree on, and how to do real-world evidence and how to transport real-world evidence to the agency that allows them to review. And you will see this on the next slide, what the process is that we have developed with the regulators. Next slide, please. You see that from the left to the right, there are certain questions that you have to ask yourself as the party producing real-world evidence. Are the data fit for purpose? And can these questions in principle be answered with real-world evidence? You will write a statistical analysis plan. You will do a feasibility analysis. And at that point, you might still decide, well, it really doesn't work with real-world evidence. If you want to go forward, then you will register your protocol. And there's an ISPOR activity going on, re-evaluating kind of the registration of protocols and how this could look like for real-world evidence. Then you will, and this is FDA specific, submit the individual level data to the agency, they were quite clear in their communication that they want to treat real-world evidence for effectiveness questions like randomized trials. They want to see individual level data. So you submit your analysis using, in this case, we are using, of course, the Aetion platform. We're sharing the Aetion platform with the FDA. They can review all the findings themselves, but they can also reanalyze the findings in the platform and do sensitivity analysis in order to assess the robustness of these findings. Next slide, please. Now, let me go through an example and some results of a randomized trial prediction that we recently completed. If we start on the right side, we focused on the Carolina trial. Carolina is an industry-funded, industry-conducted trial comparing linagliptin versus glimepiride in patients with type 2 diabetes who had some cardiovascular risk factors. And the main outcome here is cardiovascular, so 3-point MACE endpoint, but also a whole bunch of adverse events and side effects. The trial started recruiting in November 2010 and was completed the enrollment closed in August 2018. The results were presented the first time to the public in June 10th. So we did not know until June 10th what the results of this trial were. On the left side, you see the RCT duplicate prediction, also register the clinicaltrials.gov, note down the number and read the protocol. The same medications, it's the same. We tried to mimic the exclusion criteria as much as we could in these claims databases, commercial and Medicare claims data that we had available. We worked with data that we had handily available. The outcomes were in spirit the same. Of course, we had to define them in claims data, which is always slightly different from the clinical definitions that are then validated in RCTs. The data source range that we had available was 2011 to 2015. We presented on June 7th, so 3 days before Carolina trial was presented. We submitted the manuscript in January of 19, and the manuscript was then published just shortly after the ADA meeting on June 25th. Next slide, please. So these are the findings. Let's focus on the left side, the 3-point MACE, the cardiovascular endpoints. The top is the Carolina findings with a point estimate of 0.98, so non-inferiority given the 95s, and that was their target. Below that, you see the real-world evidence prediction, 0.91, the 95s also including the one coming to the same conclusion of non-inferiority. The point estimates are diverging very little. As somebody who is doing these studies now many times, I would be quite happy, and certainly in a qualitative way, the interpretations are identical. On the right side, you see one of the side effects, severe hypoglycemia. And there again, you see at the top the Carolina trial, which shows that linagliptin has a substantially lowered risk of hypoglycemia compared to glimepiride and sulfonylurea. And what we see in our prediction, we see also a substantial reduction. You look at the axis, 0.42 versus 0.15 in Carolina. We had a slightly different definition of hypoglycemia, severe hypoglycemia with hospitalization, but the clinical interpretation is the same, that linagliptin is reducing substantially the risk of hypoglycemia in the trial as well as in the real-world evidence prediction. Next slide. Now, there, Matthew Harper from Stat News interviewed several prominent cardiologists, diabetologists, and you see a range of responses to that. So Steve Nissen from Cleveland is highly skeptical, as well as David Nathan, a leading endocrinologist from the MGH, how many times would the researchers have to replicate a clinical trial before Dr. Nathan believed the results? Infinite, he said. Basically having very little confidence. But you see the FDA spokesperson much more differentiated in their response, saying, look, this is developing rapidly. We have learned a lot with regard to methods, and they are much more open to consider that. On the right side, you see Harlan Krumholz, a cardiologist from Yale, basically saying, well, I have been doing this for quite a while and have also been reproducing RCT findings. We should do more of that, particularly in the safety space. So you see the range of responses from preeminent clinical investigators. Next slide. And very briefly, of course, we all recognize, the people on this call here, there is a divide between efficacy and effectiveness, and there are several issues that you have to address. I will not go into details here, but clearly we were very concerned about data granularity with regard to the outcome measurement, with regard to the exclusion criteria. How well could we mimic the RCTs in these specific settings? We're very worried about adherence, which is why we do an on-treatment analysis and compare that to the randomized trial, where the adherence often is 90% or higher, so that the intention to treat estimate is often very, very close to the on-treatment effect estimate, particularly for these medications. We struggle with studies that compare against placebo. We routinely do better when we have active comparators, which is, of course, also much more interesting for clinical purposes, but for regulators, we still often see placebo-controlled trials. Next slide. So what is next here? As we are doing these predictions, we roll them out as we complete these prediction studies. With regard to the 30 trials that are already completed, we have agreed with the FDA that we publish them in batches. We hope that the first batch will be published this year in the fall, early fall, hopefully, made available, and then we publish the next batches as they become available. So this is an ongoing process. Please keep tuned, and of course we will communicate with you through a variety of channels. Next slide.
Thank you, Sebastian.
Thank you.
Paul, back to you.
Thank you very much, Sebastian, and also to Jeremy earlier as well. I appreciate that. And we've got some good questions that have come through from the audience, so please do keep those rolling in. We will get to them in just a moment, and I'll read out a few at once, and our panelists can have a bit of a discussion. But let's actually open it out right now to our other panelists. We haven't heard yet from Stephen and Brian. And we've obviously had some poll results from just before Jeremy and Sebastian spoke. And we've obviously had some very interesting things that Sebastian has been pursuing there. And Stephen and Brian, I guess I'd love to just hear from you on some of the practicalities and anything you'd perhaps like to emphasize from what from what Sebastian was saying.
Sure. This is Stephen. I'll take a first shot at this. But first of all, again, I'd like to state that we're encouraged by FDA's efforts to further assess the strengths and limitations of real-world evidence, in particular non-interventional study designs as it pertains to a drug's benefit-risk profile. I think Sebastian hit upon a lot of key points, one of those being that, again, results are not going to be identical. Again, many times when we run clinical trials, in fact, those results are not identical from trial to trial. So it's good that we have a framework for looking at this. The other thing, too, is, again, of course, improving trust. So with RCT Duplicate, hopefully this is going to help establish greater trust, in particular into effect sizes that are much smaller. I think we can all, state emphatically that smoking causes cancer. Again, an RCT wasn't proving that causality, but where, again, RCT Duplicate, I think, will help us with is just getting that effect size down even smaller and having trust and confidence in those findings. And the other piece, again, that Sebastian emphasized was fit for purpose. Again, RWE will not be able to address all research questions, and we should be tailoring the design and the data to fit the question. And then last of all, of course, is that we need to make sure that people have the right expertise to understand when to use real-world evidence and how to interpret the data. I'll stop there and give Brian a shot now.
So, good morning, everyone, and thank you, Stephen, and Sebastian very nicely communicated. I agree with everything that Stephen has said, but I would just add a few other points. When we think about the real-world evidence revolution, as it is, that we hear coming forward, there's, I think, a desire for it to be immediate and right now. But I think the challenge is, if we think about the clinical trial paradigm, that's been in effect and evolving for the last, 3 to 4 decades. And so I think we as a community need to begin to figure out, can folks hear me very well? It seems like there's a lot of feedback. It seems
there
to me
is a little bit
that
of feedback, but keep going, Brian.
Yeah. It seems that, we need efforts like Duplicate to begin to highlight areas where non-randomized studies using real-world data can be most beneficial and trustworthy. And as Sebastian pointed out, there are certain circumstances where it may be much better able to solve the question, answer the question validly. I think having a framework that is more commonly understood about those situations is really important. The epidemiology community, the pharmacoepidemiology community, have been doing this sort of research for decades now, but only recently have these methods really begun to become more commonly understood. And as Stephen said, a common appreciation of the methods that are necessary to achieve high quality, valid results is going to be very important. We look at something like the Duplicate program, which is an important first step in providing some of that guidance. As we come out of that, I'm sure Sebastian and others in the community will begin to try to differentiate between those situations where we are going to have more confidence versus those where we will not. Certainly the platform and the framework that Sebastian has laid out, it embodies best practices, having protocols, having clarity about our endpoints, having clarity about the analytic methods that we're going to use, and then being able to demonstrate that and have that be reproducible. These are concepts that we've been talking about for many years, but now see regulatory bodies and others beginning to say, how do we actually implement that? I think this is important, particularly for folks in the biopharma industry, because we're looking to figure out pathways by which we have more clarity about the process and what's going to be expected. And so we see these activities as being critical to establishing sort of confidence in these approaches, when they are going to be successful, when they're not, such that we can have a more concrete conversation in the drug development space about the utility of this framework. So we look forward to this, and just like I'm sure Stephen and colleagues there do, and we thank Sebastian and others for really pushing forward in this space in a way that is going to be transparent and ideally reproducible in the future.
Thank you, Brian. Unless there's any more comments from the panel at this stage, what I'd like to do is, I've had about 20 good questions from the audience in the last 3 or 4 minutes. Please keep them rolling in. I think I'll read a few of them out now, and perhaps our panelists can tackle whichever one they see fit is appropriate to them. So, my screen has just gone and resized itself at just the wrong moment, but here we go. What barriers would you expect from the industry with regards to registering RWD studies? How will adjudication be determined and standardized when using claims data, etc.? I'm just wondering if the replication of RCTs with RWD is limited, given that most oftentimes the medication just approved takes quite some time to come to market and then get recorded in claims and other data sets. How can we interpret the difference, not only the point result, but also the wide confidence interval, referring, I guess, to Sebastian's graph there? Where do you see pragmatic clinical trials ultimately fitting into this evolution of evidentiary needs for regulatory processes? Data coming from devices such as fitness wearables, glucose meters, blood pressure meters, weighing machines, etc. Are they considered valid RWD and obviously part of the sort of work that we're talking about here? So I think I'll just pause there, otherwise that will be far too many for you guys to comprehend all at once. And I'll come back to some more questions later, so do keep coming in. Who wants to respond to any of those in the first instance?
So clearly that was a mouthful. Let me just respond briefly to get the ball rolling here. The comment on the real-world evidence, the real-world data become available only after medications are marketed. That's of course true for the majority of these use cases, which is why the FDA and the EMA are very much focusing on supplemental approvals, on broadening the indications to slightly different populations, to validating endpoints, from going from lab test endpoints of the RCTs to clinical endpoints in real-world evidence. That is probably the most important space for the regulators. There are the other use cases in the approval stage where you have single-arm trials, and then you have external controls that often come from real-world data. Anybody else wants to comment? Brian, Stephen?
This is Stephen. I'll tackle, again, lots of material here and potential questions. I'll tackle the pragmatic one, or the randomized. I'm assuming that's what they're referring to. And within Lilly, often we will think about, again, fit for purpose. What's the research question? And we will start with the base of, can we use existing secondary data in a non-randomized fashion to answer the research question? Then if the answer is no, we'll look at, could we use a hybrid design of complementing that secondary existing data with primary data? If that won't work, then we move to maybe a pure primary data collection prospective study. Then when it comes to, maybe for example, the confounding by indication is too great, that our statistical models and matching will not be able to overcome, then we actually may say randomization is a necessary component of the study design to get a valid answer. And then we would ask the question, could we answer this with a pragmatic design, or do we need a traditional RCT design to answer the question? So hopefully at least that answers the pragmatic one.
Great. Any more responses?
Well, I guess what I would just add on to what Stephen just said, which I think is a very useful framework, and I think one that many of us try to walk through, is that this notion of fit for purpose, evidence and real world data as some sort of one size fits all kind of construct. And I think Stephen and I and others may be trying to bring forward a framework where it's far more nuanced, and we are tying that to the specific question and in what situation it can be best used to support the question at hand. And I think having that framework in mind, as opposed to having a one size fits all, is exactly where we need to progress the field and the conversations around this. And certainly, Sebastian's point about label expansion, or at situations where you might use an external control arm, those will be defined based on what we understand is going on in clinical practice, the way we understand that the endpoints may be measured. And I think it's in that framework where you're thinking about the elements of study design and what are the key elements that you need to get right in order to get a valid estimate. This is the conversation that I think we're moving towards, much like we've done with clinical trials and endpoints, et cetera, for the last many, many years. We have to take that same sort of thought process as we approach these questions in the non-randomized real world setting.
And Brian, this is Stephen. To add maybe one more comment, and even to address one of the other audience's questions around, time to market, and again, uptake, and are those barriers to doing RWE? And again, at times it can be. We've had a recent example where the formularies in the US were putting our product much later in the treatment sequence. Now, our research question needed to look at people who were earlier in the treatment paradigm. So not because of our label, but because of how formularies were driving medical practice in the United States, we were unable to use a real-world approach for that question and had to implement a randomized trial. So again, fit for purpose, you need to look at, again, the components of, again, who's using it, is there channeling bias, and so forth.
This is Sebastian just commenting on Brian and Stephen. I love the way you this. When I sit in front of a bunch of trialists and FDA officers, the success there often is to have a more differentiated conversation about real-world evidence and RCT and how this all comes together. Having a more differentiated conversation about a framework that integrates both of these whenever they are fit for purpose. So exactly what you two just said.
Okay, guys, I need to interrupt you just because time is marching on. I'd like to come to Jeremy for a moment, as I understand there's a few more slides to share, and then we've had some great more audience questions, so we shall come back to those. So, Jeremy, over to you again.
Great, thanks so much, Paul. And I'll try to answer one or two of those questions that we didn't address as we go through this couple slides here. So I wanted to circle back and talk about precedence and opportunity for pharma. I'm the quote guy this morning, so one more quote. What we need is more examples. It's really important that people do these things, meaning RWE studies and demonstration studies, and that we start learning from them. So this was a couple months ago, Janet Woodcock at FDA CEDAR. I wanted to show a couple of instances where RWE has been used in FDA and EMA approval decisions. And the slide isn't showing exactly right. There's supposed to be some check marks here, but probably a export issue, but we'll make those available in the after webinar downloads. I believe that's possible, Paul. But you can see that across the board here, in FDA, in the US, EMA, and Europe, two kind of major study approaches. One, single-arm trials with external controls. Secondly, other non-randomized studies. A number of recent decisions have been made using RWE. And so I wanted to call attention to a couple things. So first of all, as you might expect, most of the examples to date are either in rare disease and or oncology, in places where an RCT was either not possible or very difficult to carry out. But there's movement certainly to kind of more common conditions as well. And also, one particular case, Ibrance, recently receiving a label expansion. It's a breast cancer drug, a label expansion to include male breast cancer based solely on a non-randomized RWE study. So again, we'll make those check marks available to you afterwards. I want to take a step back and talk about how we would have achieved these label expansions, these secondary approvals without RWE. So kind of the standard timeline here, you have a randomized controlled trial or trials, pivotal trials leading generally to an approval. And then after that approval, you might have studies to inform label expansion or post-marketing commitments or requirements like cardiovascular outcome trials. RWE gives us the opportunity to think about those a little bit differently. Wait for the slide to come up here, where RWE gives us another pathway for addressing these same questions and hypotheses. So as you saw in the examples, we can use RWE to inform label expansion. As you saw from Sebastian's discussion of the Carolina trial, we can use RWE to address cardiovascular questions or other kinds of post-marketing safety questions. These could certainly be done with clinical trials as well, but I see RWE as a very robust second option for addressing these questions. RWE also gives us the opportunity to expand the kinds of hypotheses that we can ask after a pivotal trial. So we can do things like external control arms, like we saw in the examples. We can augment clinical trials by looking at additional subgroups, maybe subgroups that are too small to study specifically in a trial setting, additional endpoints, looking to change the generalizability of the trial to different populations through adjusting inclusion and exclusion criteria. Also gives us the opportunity to take a trial that was placebo-controlled and to look at it with an active comparator. Again, as Sebastian noted, a very important clinical question that can be of great service to the healthcare community. Through it all, if we want to ensure that the studies that we do and the results that we create are looked at and used for decision-making by regulators, we need to make sure that our evidence is truly regulatory grade. And so I look at the process here as the use of real-world data, fit-for-purpose real-world data, to the points of several of the panelists in our discussion, and taking that real-world data and turning it into real-world evidence. And there's a number of steps along the way there, but I've highlighted two of them. First of all, this has come up in a number of settings, Duke Margolis, FDA meetings, and other places, where the data must be fully traceable and the provenance of the data must be made clear all the way through. So from source files to final report, each data transformation step must be made clear. No black boxes. No black boxes from each bit of real-world data that is used to generate the real-world evidence. And secondly, for a causal evidence generation process, you need to apply methods that establish causality and carry them out in a way that establish confidence. And so, ultimately, what does that mean? Regulatory grade is built on principled process, and you saw this in the discussion of RCT DUPLICATE. You heard about this in the discussion that we had a moment ago. And just a small piece of this process, it's a large process covering many steps. I'll show you that in a moment. But some considerations when building the causal analytic plan. You'll see that what this does is essentially mimic the considerations that you would take into account in a trial and think about them and frame them in the same way that you would do that in a clinical trial. So from selecting the right hypotheses to selecting a meaningful patient population, through designing a meaningful contrast, exposure versus comparator, measuring exposure status and endpoints with sufficient sensitivity and specificity, and being able to demonstrate that. And that, again, is part of fitness for purpose of the data and fitness for purpose of the question, and to be able to account properly for temporality and time. All of this has to be done with strong principled process and strong epidemiology principle behind it. So as I say, even when generating RWE, think like an RCT. So this principled process in action, this is our Aetion real-world trials offering, where I showed you a little bit maybe of that third section here, implement analyses, but it's really, as I said, a much larger process where you determine the study design, identify the right fit-for-purpose data assets, implement those analyses in a causal, confidence-generating way, and then obtaining and sharing as appropriate the results fully transparently and maybe even interactively. And I know we've been talking a lot about FDA, EMA, and regulatory environments, but this, in our experience, is equally applicable to HTAs, equally applicable to payers in the US and elsewhere, and of course applicable to publications in academic journals or other peer-reviewed locations where you want to demonstrate results that have been obtained from real-world data. So with that, I'll turn it back over to you, Paul, and thank you. If you have any further questions about the real-world trials offering, you have the email address here as well.
Thank you, Jeremy. That makes a lot of sense, and really appreciate you taking us through that journey there. I hope you took note of the email address. We can display it again at the end if need be, or we can read it out. I've had plenty of audience questions and plenty of interaction, so thank you very much, audience, for keeping engaged with this. We've got about 10 minutes left, so enough time to cover a couple more things in our discussion, I think. And by the way, to those of you who've asked, I did say at the beginning, yes, we are recording today's session, so you will be able to get hold of these slides. We'll actually send you a video recording of today, so you'll be able to listen to the audio again as well. You'll be able to share that with colleagues or simply just listen back to it yourself. I think that I imagine our panel probably have a couple of points to make on top of what Jeremy just mentioned, but I'm going to hold them off for a couple more seconds, just because I'd love to read out a couple more audience questions as well to just throw in the mix, and then we can hopefully discuss all together. I shall just get my screen sorted, and then I can read out a few more. I like this question from Andrew Freeman. Isn't this question the wrong way around? Shouldn't we be assessing how well RCT reflects real-world data? After all, it's in the real world where medicines are used. And can we combine the RWE with a clinical trial, since sometimes an RCT may not have enough enrollees? So obviously a combination question there. Considering the scenario of preparing for scientific advice today, that's EMA and FDA in particular, what would be the characteristics of the development program where the panel would recommend including RWE into the clinical development program? E.g. historical controls if planning a single-arm study. Are rare diseases more suited to including RWE? What else? I'll just read one more, I think. With regard to unmeasured confounders found predominantly in RWD, what's the panel's opinion regarding novel methods to account for these challenges if RWE uses supplemental RCTs for drug approval? Okay, so I think I've thrown enough at our panel now for a lifetime. Who would like to comment on any of those or what they've just heard from Jeremy?
This is Stephen. This is, I think, an important point, and unfortunately, it doesn't hit the audience questions, but I think it's very key here. And what Jeremy mentioned was full traceability of data. And Sebastian's talk also mentioned that FDA was very interested in having patient-level data. One of the challenges that we're having is privacy and access restrictions that may make that more difficult. We have some data partners that are fully open to providing data to FDA for review and analysis. However, we have others who are not so open. For example, we were talking to a data partner in France, and they said, well, if FDA wants to look at it, they can fly to Bordeaux and sit on site and can do their own analyses. And I mean, maybe FDA is open to that, but I also think that may be prohibitive in a efficient and effective process. And we're going to need to figure out a path forward to be able to, again, use that data in regulatory decision making. But again, my worry is that with additional privacy and access controls getting put in place globally, this is going to get harder and harder.
Yeah, I think Stephen's point is actually one of the most important points that we could possibly make in this call, which is the evolving data privacy landscape and the need to have access to data is going to be a challenge that we have to face and think about mechanisms for enabling review and enabling evaluation of real-world data assets. That is something that we're going to have to pave the road on. I would like to make another comment. So in Jeremy's presentation, he showed some of the studies and some of the medicines that have used a real-world evidence approach. One of those is Blincyto, which is a medicine that Amgen has brought to the regulatory bodies using essentially historical comparator arm against a single arm filing. And I think it's important as we stare at this situation, you know, there's a lot of context why that approach made a lot of sense. And to the question about how would you think about embedding real world evidence into a development program, in that situation, acute lymphoblastic leukemia, patients who are relapsed and refractory, you know, the standard of care had not changed in 20 years. It was basically a backbone of chemo. And so the idea that we could identify patients who were not in the clinical study, but were being treated in clinical practice, the comparability we felt at that time was going to be achieved, because there was really there was no other changes in the management of the disease. And so building a population and an external control arm to support effectively an establishment of effectiveness was something that we had, I think, a good deal of confidence in. And what I think is exciting about that story and that study is that the approach we took with the external control arm was then basically verified in the phase 3 randomized experiment. And so we got nearly almost exact estimates from both the single arm historical comparator versus the randomized experiment. And it was because of, I think, an incredible attention to detail around the population, about the ability to measure the exposures, the outcomes, and thinking through what confounders needed to be identified and measured, such that we could have the ability to generate a valid effect estimate. And so as we look at these situations and a lot of the examples that are brought forward, staring at what are the underlying ingredients, the methods, and the qualifications of the parameters will always be a first order problem when we think about real world evidence studies.
And I think thinking about some of the themes that have been discussed over the past few minutes, and going back to one of the earlier questions, Paul, about other sources of real world data, and I think the example of glucose meters or certainly smartphones and such, I think you can use those kinds of data. I think as long as they're fit for purpose, and that's kind of ultimately the test here, and that brings with it concerns or at least things that we need to think about in terms of privacy, in terms of unmeasured confounding, and really accounting for any selectivity in the use of these devices. But if these devices are fit for purpose, if you can measure the exposures, if you can measure the endpoints, if you can account for unmeasured confounding, then I think our sources of real world data that we're thinking about or have been used to date could be greatly expanded over the next number of years. And I think all of these things that the panelists and the questioners have brought up are critical to be considering as we do that.
Okay, we're fast running out of time, and I'm sure Sebastian wants to have a say as well, but this is going to have to be the final comment from everybody, if that's okay, just because we've only got 2 minutes. And I'm also going to ask a question in the background whilst you all talk as well, which I think also goes towards some of the points that you guys have been talking about in the last few minutes about barriers. So we've got a good audience still on board, still got more than 90% of people here. Let's get this question answered while maybe Sebastian, you can go first while everybody has a final comment.
Sure. So with regard to unmeasured confounding, all methodologic approaches are on the table and getting evaluated by the FDA. I think what is important to regulators is that you have diagnostic tools that allow you to assess how successful you were with your confounding adjustment. And we talk about negative controls, positive control outcomes, and combining with other data sources. That is equally important as it is to apply the newest statistical methodology. I want to comment briefly on Andrew's question, why are we comparing real-world evidence against RCTs, not the other way around? That I think is a brilliant question. And of course, we do this in the RCT DUPLICATE project only in order to illustrate when we have confluence between those two study designs. Are we able to get the same finding? And in our conversations with the FDA, the conversation very quickly flips over to saying, oh, but in real world evidence, you have endpoints that we're actually much more interested than in the randomized trials that were submitted. So I very much agree with the potential for real world evidence. This is part of the learning experience to find this overlap between RCTs and real world evidence. And I'll leave it.
Thank you. I'm just going to display the results of that poll that was going on in the background while you spoke there, Sebastian. Very useful points. Anything else from the others? And feel free to comment on what you can see on your screen as well.
So Sebastian, this finding is not. Go ahead, Stephen.
Oh, no, please, go ahead.
So these are findings that we see when we speak with pharma companies, when we do surveys. It is exactly that. There is uncertainty in the system, right? And that is exactly what these demonstration projects want to overcome. There is uncertainty within organizations. There is uncertainty on the regulatory side. There is uncertainty. What will the regulator accept? Why did they accept the single-arm trial with an external control group in Brian's case with Amgen? Why did they not accept it in a different use case, right? Understanding why that is and having a clear communication and predictability, predictable pathways is critically important. And that's exactly supported by the answers here.
Any further final comments?
Yeah, I don't know, Stephen, if you were going to say something, but what I would also add is, while we ask the question, what's the primary reason? I think it's the confluence of all of the reasons, right? And, as we talk about this, we're going to be talking about this for many years to come. There's a lot of different issues that we are going to be confronting, whether it be data quality, whether it be data privacy, whether it be methods, whether it be fit for purpose, whether it be the situation that you're trying to bring forward evidence in, whether you believe the regulators have a good view on the issues, whether within biopharma there's a solid understanding of which situations are going to be workable, and then this risk issue. So there's a lot of dimensions to this problem, and for us to think that only one of them is going to have significant impact, I think they all have significant impact. And to my earlier point, this is going to be a journey, and recognizing this is going to take time and a lot of work by a lot of folks and a lot of transparency. I think the more that we understand that and are willing to embrace that, I think the better off we will be. But I think it's an incredibly exciting time for all of us that focus in this space, because there's great opportunity for us to really advance the delivery of evidence around, in a traditional, non-interventional way. So thank you.
Thank you, Brian. We are over time, so just one sentence from each of you, I'm afraid. I'm going to have to restrict you now, Stephen and Jeremy.
Oh, no, again, thank you for this talk, and again, great job from the speakers, Sebastian and Jeremy, and thanks for the opportunity to represent industry. And I also like the optimism of the 5% full steam ahead.
And just very quickly, again, thank you to everyone. I think a really, really interesting panel, and I agree with Brian. There's an incredible opportunity for us ahead, and look forward to really diving into some of these issues that we've talked about.
Indeed. Well, yes, thank you to all of you as well. Huge thanks for sharing your time and energy with us today. Some genuinely fascinating insights. And of course, we still have many audience questions left unanswered. So I haven't asked permission from our 4 panelists, but I will be asking them after the event whether or not they'd mind spending a little time actually answering some of those. And we'll obviously do our best to get them those replies sent back out to you. And hopefully that can color in any missing gaps for you. Really, really appreciate you all being so much part of this webinar and making it interactive. I'm actually going to hold the webinar open now, even though I'm going to stop talking for a couple of minutes. That will allow you to please ask any more questions you have, or maybe send a message of thanks, or maybe send a message of a suggestion of where you think we can focus going on from here, what unanswered questions you still have. So please do send that in before you depart from today's session. In the meantime, really appreciate everyone that's on this webinar. A very quick notice that if you want to carry on this conversation, you very much can do. 2 conferences not happening until next year, but please put them into your calendars now. We're going to be continuing this conversation there. Hopefully it will have moved on in the months between now and then, and we'll have quite a different conversation when that comes up. I would love to wish you all the very best. Again, another particular thanks to Aetion for helping us assemble today's webinar and make sure that it indeed was engaging, and very clearly was, given the audience response. So thank you so much to Aetion, thank you so much to our panelists, and thank you so much to you. Have a great day, and hopefully see you in person next year. Thanks very much. Bye-bye.